you very much for the introduction and for the invitation. I will talk about metagenomics, basically, microbial dark matter here in this case, and how we can use cloud computing resources for big data analysis and the combination of these two. I was expecting a few people who don't know what DNA is, but so when I talk about data today, this is the data I'm talking about, okay? So basically these are large strings of 4 letters, and we want to analyze these strings, DNA of different organisms. When you look at the history of DNA sequencing and how it emerged over the last years, and you see that there's a huge or tremendous advancement in the technology of DNA sequencing. So this slide here that starts in the year 2000, that was when the human genome was finished, the first human genome, and that was done by a technology called ABI sequencers, Sanger technology, and these sequencers could produce about 5 kb per day, 5,000 bases per day. So overall, it took about 10 years to complete the human genome with thousands of collaborators internationally, and the whole project costs about $100 billion to sequence just the single genome. And you can see over the years there were different technologies that turned up. There was a 454 that could sequence 400 megabases per run. Then there was Illumina with 15 gigabases, and the cost dropped exponentially, so there's an exponential scale on these axes. And now we're actually at a price where we can sequence a genome for about $1,000, right? So it is a diagnostic basically today. So it's cheap enough to do this on a regular basis. And the machines generate about 6 terabases per run. Okay? And of course that's a problem if you want to analyze it. At least for most people it's a problem. Because how do you do the analysis usually? When you are computational scientists, what you do is, here's a scientist, he wants to analyze the data, right? So what you do is he's working on, let's say, a laptop. He pulls down code from code repositories like GitHub. He pulls down databases. He wants to compare his data against 2 local resources, right? And then either he can do the analysis on a local workstation, or if the data gets too large, he has to go to a larger compute cluster. But if you look at the amount of data that's available in these public databases, you see that the amount of data is also growing on an exponential scale, right? So today we are up to like 12 petabytes of data which is available, and if you want to do large-scale analyses on these datasets, it takes a long time to download the data, and probably you don't have the resources available. So this is just an overview of what kind of data is available in these databases. And as I'm going to talk about metagenomics, just to get an idea, there are metagenomics data sets which have in total about 250 terabases of data. Okay? And I will come back to this point later. And as I said, that's a problem. For many researchers, that's a real problem because they don't have local resources to use or to do the analysis, basically. And this is where cloud computing comes into place. So cloud computing addresses these needs by providing scalable services. Scalable meaning scaling up with larger machines or more cores, or scaling out with just more machines. And then hopefully you can do the analysis on a larger scale compared to your local resources. And of course, if you pool these resources, there's also these economies of scale, because you have access to more resources for, let's say, a short time, and the resources are really utilized over time. So that's also a nice advantage of pooling these resources. For scientific applications, it's very nice to have these virtual environments. Virtual environments could be virtual machines or Docker images or singularity environments, which allow maximal flexibility. So you are completely free from the underlying operating system in terms of software requirements or dependencies on your operating system, or special software libraries, which is usually a problem when you try to run your install libraries on a shared compute system. That's always a problem. So these virtual environments, you're maximal flexible, you can install whatever you need. And the nice thing is you can also share these environments, and which gives the guarantee that the research is actually reproducible. So you can share the virtual machines, you can share the Docker containers with your colleagues, or even in a publication, and that makes the whole research reproducible. Okay, so where do we get the cloud from? So luckily there is this German network for bioinformatics infrastructure. It's a German-wide network with about 40 project partners in 30 institutions. There are 8 service centers, and the de.NBI network is the national German node in the larger European bioinformatics infrastructure, which is called Elixir. And the mission of de.NBI is to provide service to life science researchers, also training, and it is basically the hub for cooperating with international institutions, just as I mentioned, the Elixir. And de.NBI also provides a cloud environment, the de.NBI Cloud. It's a federated cloud in 5 locations in Germany. And the services which are provided are on different levels. So it could be infrastructure as a service, where you just get access to the infrastructure and have to set up everything yourself. But it could also be platform as a service or software as a service. The cloud is funded by BMBF, just as the whole de.NBI network. So there was a huge investment in the infrastructure with 2 rounds of funding. The first funding was for compute resources. The second funding was for storage, just to get access to large reference data sets. Altogether, the cloud has about 15,000 cores now, more than 14 petabytes of storage. We have high memory nodes for special applications, up to 3 terabytes of RAM for, let's say, assembling large genomes or metagenomes. And there are also a couple of GPU and FPGA nodes available. It is free for academic research, so everybody can use it for free. And you get access through the Elixir AAI, which again is connected to EduGain, OrchidID, or Google. So once you have registered, or your university is part of EduGain, it's really easy to get access to the resources. If you want to run your project in the cloud, you have to fill in a project or apply for resources, basically. It's a very simple project. You send in your project application, which is reviewed by a committee, by a scientific committee, and after approval you get access to one of the cloud sites and can start working on this. These are the numbers of project applications since the official start of the cloud. So that was end of 2017 when we started making this publicly available. And as you can see, it's really nice that more and more people are using it. So we have about 100 project applications or projects on the cloud now. There are more than 300 developers in the cloud, and there are over 13,000 cores already allocated for these different projects. What we are planning are large mirrors of huge data sets. So for instance, the ICGC mirror is a mirror of like cancer data, which is, I think, about a petabyte of data, which will be installed in at least 1 or 2 of these centers, so that people have direct access to these large data sets. When we started working with the cloud here in Bielefeld, there was actually a couple of years ago when we started using the Amazon cloud, because there was no other cloud available. We thought, well, how do we make this easily accessible to users, right? Because usually you just start a VM, or 2 or 3 or 100 VMs, and then you have 100 VMs which are not coupled in any way, and you have to set up some sort of compute environment. And this is where we came up with this what we call BP Grid, cloud computing platform, which sets up a compute environment which basically looks like your local HPC environment. So if you're familiar with HPC environments and then queuing systems and so on, then it's really easy. It's just a simple config file where you specify what kind of frameworks you want to have installed on the system, and then it will just launch the VMs, configure the VMs, you log into to your cluster and start distributing your jobs on the cluster. The BP Grid is, of course, compatible with de.NBI Cloud, but you can also use Google or the Amazon Cloud, whatever you want to use. And after the second round of funding, which I just mentioned a couple of minutes ago, we thought about what kind of data do we want to provide as reference data. And as we do a lot of metagenomics here, we thought of, well, let's start with metagenomics data, raw metagenomics data, because we had some nice ideas of like how to use these raw data sets. So the goal was to mirror the metagenomic data of the short read archive, basically, the ENA data sets, into the de.NBI Cloud here in Bielefeld. So luckily there was a grid FTP server available at EBI, and we started downloading the data sets, which was about 270 terabytes of data in 190,000 FASTQ files. And it took about 3 weeks just to transfer the data to our local storage. But now this is available locally, and I will show you some applications how you can actually use the local data to well, maybe mine public data sets for your research. One example I want to show is this one here. There was a project I started when I still was at the JGI. It's called the Microbial Dark Matter project. And what we try to do is to illuminate the tree of life. What does it mean? Well, when you look at the phylogeny or the taxonomy of life, the tree of life, you see the archaea here, and these are the bacterial genomes. And what's shown by the colors is, okay, we're looking at the phyla level. Okay, phyla level means, so if you're not really familiar with taxonomy, phyla level means we are looking at this level here, right? So there is life basically, which you can classify in different domains, kingdoms and phylum, and down here are the species, and these are actually the genomes we can sequence, right? Like when you start sequencing an organism, you're always sequencing a species. But of course they are related to each other, right? So the species are related to each other as a genus. These are related in a family, so there's a hierarchical tree basically, and we are looking at the almost the highest level of the tree right now when we look at this tree. This is a phylum level tree. And when you look at this, the gray branches here, the gray branches are the phyla which are where we have cultured organisms, right? And by cultured organisms, I mean you have organisms which you grow in the lab, and when you grow them in the lab, it's easy to sequence them. Okay? So for these gray branches, we do have genomes available. But for the black ones, there is no genome available in any database. Right? We know that they exist based on marker genes we can sequence, right? Like a 16S gene. We know that these black branches here, the black phyla, exist, but no genome sequence is available for that. And that was the goal of the project, to get genomes for these dark branches here. And when you look at the databases and the distribution of the genomes which are available in the database, then at that time, that was in 2013, 87% of all sequenced genomes belong to only 4 phyla, right? So there's a huge bias in the database. And if you want to do like a larger scale comparison of different genomes, of course you are always struggling with this bias because you only have these phyla sequenced in the databases. Okay, so what do? You talk to colleagues, and this is what JGI did. This is the team of the whole project. So it was initiated by JGI. and you send the people to weird places in the world to collect samples, right? And in this case, these are the samples, 9 different sampling sites. There are wastewater from acid mine drainage. There are lakes, very salty lakes here. There are deep smokers from the bottom of the ocean, and so on. So it's a very, very weird places where they took samples. And then there are basically 2 different approaches how to get at the genomes. Right? So you look at the microbial community. When you look at the community, you can use these approaches to actually sequence these. You could do a metagenomic approach, which means you just extract all DNA from these sequences, or from these organisms, sequence the DNA, and then analyze it in some way. You can try to enrich it. Right? I mean, that doesn't really help if you want to target particular branches in the tree, right? Because there still might be a bias, and you don't get the phyla you're interested in. So you can try to enrich certain organisms, or the other approach would be to use single cell sequencing. And single cell sequencing works like that. So you basically you isolate the cells. So you're looking at a single cell. And then from a single cell, you isolate the DNA molecule, which is inside the cell, and then you have to amplify it, and you can sequence it. That sounds pretty easy. It is actually a very difficult job because, of course, you're always struggling with contamination in these samples, right? Because there's not only one cell. There might be free-floating DNA in the sample, and so on. So it's a very difficult kind of analysis. But anyway, so in this project, they got almost 10,000 cells. They amplified the cells, so they ended up with 3,000 cells. From these 3,000 cells, they sequenced the 16S gene first, and based on the 16S gene, they know what kind of phylum it is, and then they sequenced 200 single cells deeper to reconstruct the genome. And then when you map these genomes back to the tree of life, you see that now the 200 genomes actually nicely represent the tree of life. Okay. And you can also look at the biology, of course, and that's actually something you want to do. First of all, you can look at the tree again, and it turned out when you create a new tree based on these 200 genomes combined with 2,400 species from the database, there is actually some reshaping of the tree in some of these parts of the tree, right? And that's just because you have more information available now, more genomes available, and the resolution gets better, and you can get a better view of parts of these trees. Okay. Generating these trees is actually not easy. It took about 130,000 CPU hours to generate these trees, right? Because usually you do this multiple times, and with these many species, it took quite a while to generate this, right? 130,000 CPU hours. If you want to run this on your laptop, you have to wait for a long time. Of course, you could use the cloud, right? At that time, we could have used the Amazon cloud, right? If you want to do the same analysis on the Amazon cloud, you pay about $11,000, right? So you can easily, well, you rent 1,000 CPUs on Amazon, which costs you, let's say, 10,000 euros, and you get the analysis done in 130 hours. Okay? You don't have to buy your machine; you just rent it, shut it down, and that's it. Right? And so that's an easy thing to do. You can also look at the functional novelty of these genomes, and I just want to point out just 2 examples here. So what we found is a recoded stop codon, basically. So there were organisms where the stop codon was actually coding for a particular amino acid. Right? That changes well, if you run a regular gene prediction on these genomes, of course you would have problems because your gene prediction would always stop at the stop codon. And if you don't know that it's actually recoded, all of your genes are too short, basically. Right? So this is just one thing we saw. We saw horizontal gene transfer from eukaryotes to archaea and so on. So I will not go into detail here too much. And of course you can use the data now to what's here called phylogenetic anchoring, which is basically a fragment recruitment. And what you do is you take, when you're interested in, let's say you sequence a metagenome, right? You just get random reads and you want to know what kind of organisms are in my metagenome. You can take a number of reference genomes and map the reads back to the reference genomes. Right? And as I said, there was a huge bias in the database, and if genomes are missing, reads might be classified to the wrong organism, right? Because the best match, you always take the best match, right? And if there's a big bias in the database, then these matches, of course, are wrong. So what we did is we added these 200 new genomes to the database and then did an analysis of 475 public metagenomes, which was a pretty high number at that time, and ran some blast searches, and you can see like what the rate of like newly annotated, newly taxonomically classified sequences you had in your data set, right? So for instance, when you look at this sample here, Sakinav Lake, like 20% of the reads of this metagenome were now assigned to one of these or some of these new genomes, right? So you have a reclassification basically of 20% of your genome, of your metagenome, right? And this is so as you can see, these red dots means okay, there's more than 3% of reclassification. For some of them, there's not a big difference, but I mean there is a difference now. Now that the genomes are actually available. So there are weird reactors, like this is a waste reactor, I think, where 11% of the reads were reclassified. This is the Global Ocean Survey, 6% of the genes or the reads could be reclassified and so on. So it's a very valuable resource when you add these genomes to the database, and because now you can do a much better taxonomic classification. But of course you can also do metagenomics, like a whole genome shotgun metagenomics approach, when you want to reconstruct these genomes, right? So now what you do is, again, you take your metagenome. This is a different study here, but anyway, you take a metagenome from some environment and start sequencing the DNA, right? Just random pieces of DNA, pretty short pieces. And then you do what's called an assembly, and you end up with longer stretches. And how does this work? Well, you have to think of a jigsaw puzzle, basically. When you do a jigsaw puzzle, this is basically what you do. You run an assembly. So this is an easy puzzle here, because in this case, the reads are pretty long, or the pieces are pretty big, right? And when you reconstruct the whole picture, do you recognize what it is? Probably, this is easy to do, okay? If you have two genomes in your sample, that might still be easy to do. Right? So now you have two genomes. Of course, you have to imagine that the pieces are all mixed together. But anyway, I think in this case it would be easy to reconstruct these two genomes, right? And why is it easy? Because they are pretty different, right? Unfortunately, genomes are not always different, right? There are genomes which are very similar to each other. They could be similar, like even within a genome, they could be very similar, right? So if you think there are like 1,000-piece puzzle here, or even a 1,000,000-piece puzzle here, it might be difficult to put pieces of a puzzle like here or here or here or here, because you have repeats of these structures, right? And that's the same. That's the same in biology. So it is a challenge to even reassemble like single genomes, right? And then even like larger ones could be similar but not the same. Okay. And if you look at the metagenome, the problem looks more like this, actually. So for each of these houses, remember, just imagine you have for each of these houses, you have a jigsaw puzzle, and now you mix everything together and try to reconstruct it. And that's basically what metagenome assembly is. But amazingly enough, that it works, sort of. So you get larger pieces, but you still don't get complete genomes out of it. But anyway, you can take these longer pieces. What are these are called contigs, contiguous sequences, these longer sequences here. And you can try to do a taxonomic binning of these. So basically you want to group everything together which comes from the same genome. Okay? And then you assign them to particular taxa, and whatever has the same color here belongs to the same genome, and that's the way you reconstruct the genome, start annotating the genome and look at genes and function and so on. So how is this done? The way it's done today is basically you look at the sequence again, you look at the formers of the sequence, all four combinations of the four letters, and you just count the frequencies. Like how often do you see AAA, and how often do you see AAAT, and so on, right? And then you can use these vectors, basically frequency vectors, and start clustering it. And this is how it looks like when you cluster it. And what you see, there is a structure in the metagenome already, right? So each of these clouds here is actually a genome. Okay, so it works. That's nice. Unfortunately, when you look closer, it doesn't really work because some genomes are really similar to each other, right? Just as the buildings I've shown before. And what you see now is the different colors here meaning different genomes, basically, and the different genomes are clustering in the same cluster. Right? But how can you split these into 2 or 3 different genomes here? And the information you use for doing a subcluster basically is the abundance of a genome in your sample, right? So the genomes itself, they are very similar to each other, but you can look at the abundance, like how many of these cells do you have in your sample, and then you can use this additional piece of information to recluster this, and then it actually works pretty nice. This is just to illustrate this is another example here. So each dot here, which you can hardly see, but each dot is a contig. The contigs are colored by their GC content, which is related to the tetranucleotide frequency I just mentioned before. And on the X and Y axis, you see the abundance or the coverage of these genomes in your data set, right? And this is a replicate. So they should all be on the diagonal. So there are 2 samples which are technical replicates. But if you take 2 samples which are a little bit different, let's say you do a time series or something, right? It might look like this, right? And you see that there are sort of clusters here, but you don't really know if they really come from the same genome. But at least if the color is similar, it means that the GC content is similar, which could be an indication that this is actually the same genome. And when you try to visualize this a little bit better, you see that these clusters are actually sticking together, right? And so each of these are now actually genomes, and when you change the coverage, these clusters stick together, and this is an indication that this is actually one gene. Okay? And now, and from like large metagenomes with many samples, you can reconstruct a lot of genomes actually from metagenomes. So this is an example of a mouse gut metagenome. And for the mouse gut metagenome, we got 740. Sorry, we got 740 genome bins, which were at least 50% complete. Completeness you can estimate by looking at marker genes, which I will not go into detail here, but there is a way. What's going on? There's a way to estimate the completeness, and there's also a way to estimate the contamination. You can see that there are like 400 genomes which have like very high quality from this data set. Right? So you can reconstruct these genomes from metagenomes without doing any culture, just doing metagenome sequencing and reassembling. So how can this be used? This approach, this is a different project now. So I'm showing 2 different examples now of projects we are currently working on. This is a Virus X project. This is a project of viral metagenomes, which is only trying to target viral metagenomes. And the idea of the project is basically bioprospecting. There are companies involved in the project which are looking enzymes which are interesting for industrial applications, basically. And they look at very weird places, these viral metagenomes, and we're trying to analyze the metagenome. So basically here they take the samples from different habitats. There's this big block of bioinformatics, which is basically I will show you the pipeline. This is the assembly, the annotation, and so on. And then, of course, there are people trying to clone and express the genes, and then at the end, even for unknown genes, try to predict function of these genes by doing 3D crystallization and so on. So it's a huge project. First thing you do is you go sampling. This is a sampling trip on Iceland. It's a hot spring, and you can see that the water is actually pumped into these containers, brought to the lab. DNA is filtered out and then sequenced. And there are other project partners which also go to very weird places. These are these dark smokers, hydrothermal vents in the ocean, where they take samples, and we sequence the samples and analyze the samples. And for analyzing the samples, we've set up pipelines. Christian, sitting in the back, has set up this pipeline of different tools. So each circle is basically a tool here. And you see the data flow within the whole pipeline. Okay? And each of these circles can actually be another pipeline which is integrated in this circle. So it's pretty complex and nice pipeline, which we can run in the cloud now because everything is automated, right? And we can run this on large data sets. And why do we want to do this? Well, of course, we want to know. Let's say there are lots of unknown genes in these viral metagenomes where you have no idea what the function might be. Okay. So the idea is take the known viral sequences, take the data set from the project, but also take the sequence space which is available in public data sets. Assemble all data sets which are available and then integrate the data, right? So take the project databases, take the assembled data sets, and then run protein clustering tools to see if something clusters together and so on, just to get an idea of the function of these particular proteins. So Christian ran the analysis on, in this example, 2,300 publicly available metagenomes. I think now we are up to 4,000 public metagenomes that again took about 40,000 CPU hours in the cloud. We ended up with 140 gigabases of assembled sequence and 248 million predicted genes. Right? And now the goal is to cluster these genes and then look at clusters and see if there are actually genes which might be of interest for our commercial partners. Okay. And you also get these nice interfaces to look at the data and so on. So this is just one example how you can make use of these data sets, the public data sets, and integrate them into your analysis just to with more data, hopefully get a better understanding of what you're looking at. And then the last example is this one here, SparkHit. This is a platform for large-scale analysis, again, of these next-generation sequencing data in the cloud. So it's a cloud-based platform. What you do is you set up a large number of VMs, called worker nodes here, and then you distribute your data on these worker nodes. And what the program can do is it can very fast, it can map reads to a reference genome. So think about these microbial dark matter genomes, and you want to know where do these genomes occur, right? Just as the example I showed before. But now we don't have like 470 data sets available, but we have like 100,000 data sets available. So you need the tool which is really fast and can make use of these resources. And that's actually SparkHit. So it takes the data sets, the raw data, it maps it to your references, and then you can generate some sort of profiles for the references. This is a benchmark. The benchmark was actually run, I think, on, yeah, that was run on Amazon, in the Amazon cloud. So it takes a while to set up the cluster. It takes like up to, for 100 nodes, 32 cores, 3,000 cores basically. It took up like 20 minutes just to set up the cluster, but afterwards you could analyze 8 terabytes of data in this case. This is human microbiome data. You can screen 8 terabytes of data in less than 2 hours basically. So it's a very nice tool to do some sort of high throughput screening just to see where particular reference genomes are available in your metagenome samples. And then the output looks like this. So these are different body sites here. So that's the ear, the nose, the mouth. These are the reference genomes we're interested in. And so, for instance, if you look at this one, this is E. coli. Where should it occur, or where would you expect it to occur? Of course, in the gut. This is a gut sample here. And what you see is that there are a lot of sequences. This is a y-axis, a number of hits basically you get, and the identity of the hit. So it's 100% identity. Many sequences have 100% identity to the E. coli, just as you would imagine, right? But there are also examples like this one here, for instance, Streptococcus mitis, which occurs in the mouth somewhere, but it's not 100% identical. So there seems to be a related genome, right, which only has like I don't know, 90, let's say 90 or 95% identity in this particular sample. So you can do a high throughput screening and see where your genome is present actually, and use this for other purposes, let's say. Okay, so where are we right now? And again, this is actually a picture which is a couple of years old, but anyway, I think it hasn't changed much. What are you looking at? Well, the blue genomes here, right, are the genomes which were sequenced with regular sequencing projects, let's say, microbial genomes, okay? So at that time, that was in 2009, I think, or 2000, actually that's a paper from the 2013 Microbial Dark Matter publication. There were about 2,500 genomes available in the databases. And what you see on the y-axis is the phylogenetic diversity, right? Like how diverse are these genomes? And you can see there are, well, there is not a lot of increase anymore. So people are sequencing genomes and genomes and genomes, but the phylogenetic diversity doesn't change. Right? So there was an initiative, the GIBA, Genomic Encyclopedia of Bacteria and Archaea. So there was an initiative to actually increase the diversity of cultural organisms. And you can see these about, I don't know, 300 genomes or so, they increase the phylogenetic diversity a little bit, right? And the red one are these 200 microbial dark matter genomes, right? And they, again, increase the diversity. But if you look at the known diversity, and the known diversity is this one here, right? This gray one here. And the gray curve is based on the 16S genes again, the 16S marker genes, and the diversity is much higher. It's in this case, it's going up to 5,000 in the green genes database here. And we are down here, right? So there's still a lot to do to explore the whole tree of life if you really want to shed light on each branch where no genomes are available so far. Okay, so these are the people which are involved in the different projects. So there's an de.NBI Cloud team here, together with a CBTech Bioinformatics Resource Facility. Jan and Alex and Christian helped a lot. Peter Bellmer is the cloud governance, responsible for coordinating the cloud and the applications and so on. BBGrid was developed by Jan and Christian and Marcel and still in development. The Spark Hit was developed by Liran. And yeah, that's it. So thank you very much for your attention.